Yeah, so first of all, I would like to thank the organizers for giving me the opportunity to speak. I am a PhD student in Stefan Gruber's lab in Lausanne. And today I will show you some of the yet unpublished data on whether SMC complexes meet on the bacterial chromosome. And if they do, what happens then upon the encounters? As you have already heard several times during this meeting, SMC complexes are highly conserved proteins that are important for organizing the chromosome, and they do so presumably by loop extrusion. And the ability to extrude loops was already shown before in vitro experiments for some of the complexes from the family. However, what happens and if they meet on the chromosome, what happens then? This was not yet well described. And we can envision several scenarios when this happens. That one complex can bypass through another. One complex can push back the other one, or also we can imagine that the complexes stall and fall off the chromosome. In our lab, we use a bacterium, Bacillus, for our experiments, and I would like to give you a few facts on its chromosome biology. So Bacillus has a single circular chromosome that is 4 megabases in size, and in contrast to eukaryotic organisms, many bacteria, they have specific loading sites for the SMC complexes. And those loading sites are localized close to the origin of replication. And they are, in most cases, they are closely juxtaposed. However, within, let's say, 50 kbs from the origin. However, in Bacillus, they are more sparse within 800 kbs, close to the origin. And also what is important to mention here, the parasites, the loading sites are marked with triangles, and they have, according to chip experiments, PAR-B chip experiments, they have different affinity for PAR-B. So how the loop extrusion complex is loaded, there is a loading complex for SMC, the PAR-B, that recognizes the loading sites and then recruits SMC there. And next, SMC actively translocates to the flanking region, ultimately forming a gradient of SMC enrichment from the origin towards the terminus. And as a consequence of this, the right arm of the chromosome is closely juxtaposed with the left arm on the chromosome, which you can also see here on the bottom left. And this we can observe in chromosome conformation maps. And here you have a wild-type example for Bacillus subtilis. In all my maps, the origin is placed in the middle, the terminus is at the extremities of the map. And you can see here that there's a very prominent secondary diagonal showing increased contacts between opposite arms of the chromosome. And SMC complexes have a very unique architecture. And one prominent feature is this long coiled coil that we do not know much what is the role of this coiled coil. And I took advantage, cut the long story short, and not getting too much into details how the constructs were made. We have a collection of resized SMC complexes that have either elongated or shortened coiled coils. And I have selected the complexes that are functional. They support growth of the bacteria as in wild type, and they are expressed to wild type levels. And I investigated whether changing the coiled coil length has an influence on chromosome organization. And so here again, you can see the wild type map. And we were very surprised to see that when the complex was modified, either elongated or shortened, we lost the secondary diagonal. So the arms could no longer be juxtaposed. However, there are some contacts, but they do not extend over the origin region, which would suggest that there is some kind of a barrier in this area. Interestingly, when we inactivated most of the PAR-S sites, the loading sites, and limited it just to a single one, then the chromosome alignment was rescued, at least to some extent. So this was very interesting for us. And then we wanted to know what is the distribution of those complexes over the chromosome. So here you have chip-seq profiles, and you can see that the elongated, the modified SMC complexes, they accumulate at the origin region, in contrast to a shallow gradient that you can see in the wild-type situation. This scenario of high enrichment over the origin could be rescued by limiting the entry gates of SMC for SMC complexes to a single one. So what we think is happening here is that when the modified SMC complex, why is it accumulating at the origin? We think that given that in the cell there is a limited number of SMC complexes, the elongated complexes must be falling from the chromosome and be constantly enriched, ready to be loaded at the origin instead of traveling all the way to the terminus. And so what we imagine is happening in the wild-type situation is that the complexes are known to have, in bacteria, have a high residence time on the chromosome. So once they are loaded, they travel towards the terminus and only get unloaded there. In case of modified SMC complexes, their stability on the chromosome is influenced, reduced, and so they fall off and increase the pool at the loading site. So we were wondering whether we can force the wild-type complexes to do the same, to clash and fall. And to do so, we did two experiments. We thought about two experiments. One of them included limiting the loading sites to 2 and then varying the distance between them. And the second approach was to increase the amount of SMC complexes and observe what happens then. So for the two identical loading sites, we've picked one of the par S sites that was shown in chip experiment to be very efficient in recruiting par B. And I introduced the sequence at other locations on the chromosome, either on the right arm of the chromosome or on the left arm. And here's what happens when you have a single loading sites. So all the introduced par S sites, they could be the inflection point for chromosome folding. But when we combined two identical loading sites, we saw new patterns appearing. So here you can see that there are two parallel secondary diagonals that originate from the par S sites, and they become, after the complexes meet, after the meeting point, they become more diffuse. And this becomes even more pronounced when you move the par S sites further apart, being consistent with the notion that when the SMC complexes have more time to extrude the loops, then problems arise. And the other experiment was increasing the pool of SMC complexes. So here we introduced a second copy of SMC, and we got 4 times more SMC complexes in the cell as judged from Western blot experiments. And we assumed that if the complexes were to bypass, then we would not see much difference in the chromosome conformation maps. However, if they cannot bypass, then the known chromosome architecture will be perturbed. And this is exactly what we saw. So when you have 4 times more SMC complexes, the secondary diagonal is getting more diffuse. And consistent with this notion that if you reduce the amount of loading sites, you rescue the chromosome. This is exactly what we saw when we just left one. So to summarize, I would just like to say that even though the bypass strategy is, we cannot exclude it, we think that this is rather inefficient in a wild-type situation for the cell. And we think that the cells would rather avoid collisions, and by limiting the amount of SMC complexes available in the cell, and with the notion that they have a high residence time on the DNA, and also by clustering the loading sites so that they limit where the SMC complexes are entering the chromosome. And with that, I would like to thank my lab and also our collaborators in Suribet. And I would also like to thank you for your attention, and I am happy to take any questions.
Thank you very much for this nice talk. Yes, there are some questions. Daniel, you have a question?
Hi, Hannah. Great talk. So it looks like your data are very close to the one that are from the group of Wang, the Wang groups, right?
Yes.
It's a different experiment. I mean, it's a different experiment. Is it? I mean, your results are similar to them, or you see some differences?
Well, yes. So the experiments are pretty similar, but what they did not see, if I remember everything correctly, they did not see the two parallel diagonals, and we see them. Of course, the conditions in which we do the experiments are slightly different. We do not, these are exponentially growing cells. We do not stop replication or anything. So this might be one reason why they are slightly different. But yeah.
And from your data, are you able to somehow infer what is the probability when two condensing meets? What is the probability that they cross each other, or can you estimate that?
Well, we would love to estimate that, but we don't model the data, or we are not very strong in this area. But we would be happy to have this kind of a collaboration.